Bismillahirrahmanirrahim. I am Falak Fazil and welcome to Alpha Genomics official YouTube channel. In today's session, we will discuss about flow cytometry, the technique, the principle, and how it can be used in our research. So, just giving you a brief introduction about flow cytometry. Flow cytometry is a laser-based assay or technique that is used to study the chemical, the physical properties of a cell that is maintained in a suspension. So the principle of flow cytometry is based upon single cell analysis. Why, as a researcher, I prefer flow cytometry over other techniques, as it is a recent and most advanced technique. And the second thing is that rest of the techniques normally go with the principle of pooling assays. Flow cytometry is a single cell technique that is used to study the possible occurrence or possible functioning of a single receptor over the cell membrane. Now, in your research, in what kind of activities you can use flow cytometry? Flow cytometry in research is used to determine the cell characterization, the cell sorting, determining the cellular function, and to detect any other microbe or any other microbial or fungal material that is in suspension, and to study the biomarkers. In this biomarker, normally we deal with the immunophenotyping of the immune pattern cells, which is used to determine the concentration or the expression of certain antigens in the peripheral blood. It can also be used for the prognostic and diagnostic effects of your blood or your bone marrow. Now, when we talk about flow cytometry and when we talk about the single cell analysis, currently at Alpha Genomics, we are running few assays that are our regular practice. They normally include immunophenotyping, as I've already mentioned. We deal with the myeloid markers, a complete set of myeloid markers that are based on the identification of certain antigens. And along with these markers, we usually perform the cell viability assay. That includes annexin 5 and propedium iodide. Now, how can this cell viability assay be different from, or this flow cytometry can be different from the rest of the trivial assays that are the serological assays? Normally, the serological assays are based upon the impedance value. They do give you a net count of the cells that are present in your peripheral blood, but among this cell, how many of these cells are in any necrosis phase, how many of the cells are live, and how many of the cells are at any apoptosis phase is determined by using these specific antigens, and how many normally these cells are given in the form of percentages. So, in flow cytometry, we deal with the multicolor staining. Multicolor staining is a visual display of the cells by staining them with any fluorescent dye. There are three different channels on which these dyes fall. They can be 4 or multiple. Currently, we are dealing with 3 different channels. Now, here comes a question. For the assessment or the drug development assays, you normally use for the cytotoxic screening of the cells, or for detection of any antimicrobial or any antiviral activity of the cells of the particular drug, you deal with this cell viability assay. And for the effective treatment, like we have a broad spectrum of people working on leukemia, working for the drug development of leukemia, working for the effective treatment of leukemia. Even currently we are working for the pharmacogenomics stuff and for the precision medicine. So the count of these particular antigens, which normally include CD8, the T helper cells, CD19, the B helper cells, CD4, the T helper cell, and CD14, the T helper cell, B helper cell. So there is an amalgam, or the specific markers are present in our body or in our bloodstream to detect the particular immune response. We even deal with the stem cell marker that are commonly used, like CD73, CD90, CD105. So the marker can be dependent upon what kind of study you want to do. It's not like a hard and fast rule that you should deal with only these markers, but these markers can vary according to your study. The most important thing while flow cytometry performing this technique is the availability of the cells in a single suspension, single cell suspension. The single cell suspension, as it is the basic principle of the instrument, so it's really important to prepare your sample in such a way that each of the individual cell is separated from one and the other. In case of the peripheral blood, it is easy to separate the cells, but when you are analyzing a tissue, it is quite difficult. So for the analysis of certain tissues, you normally use or require the ultrasonication for the breakage of any adjacent proteins that are holding the cells one along with one another. Today, I'll explain you a brief introduction about the WBCs, the immune pattern cells. So this is a vial, you can see a 50 ml falcon. I've already separated these cells. The cells, your immune cells. I have separated the immune cells by using the technique of density gradient method. And for the density gradient method, we normally use lymphocyte separating media that particularly targets the lymphocyte. It provides the cells according to the density, but the prime interest of the cells are the lymphocytes over here. So I have prepared the cells in a suspension and maintain them in suspension. Normally the suspension buffer we use for the stabilization of these cells is PBS buffer. It helps us to give a cell an environment that protects it from any degradation, from any death or any external torture. This is all for today's session. We'll continue the sequel of this training series or this current training regarding flow cytometry in our next session, and I'll explain you people about the working of flow cytometer, the troubleshooting of flow cytometer, and we'll practically perform an immunophenotyping assay, and the next session would give you a complete review, a complete description of the staining processes, the gating processes, and rest of the stuff. So thank you from my side. Stay connected and stay tuned for the next session.